Okay, so welcome back to this next video in which we are discussing the voltage-gated sodium channel. Okay, right, so we've discussed now the structure of voltage-gated sodium channels. We've discussed that they consist of this alpha subunit, of which there are 9 different forms which have differential expression. We've discussed that NAV 1.1, NAV 1.2, NAV 1.3 and also NAV 1.6 are found within central nervous system neurons, whilst NAV 1.7, NAV 1.8 and NAV 1.9 are found in peripheral nervous system neurons. NAV 1.4 then is found in skeletal muscle cells, and NAV 1.5 is the main form found within cardiomyocytes. Okay, then once you've got your alpha subunit, you then have the option of adding up to 2 beta subunits on. So there is one attachment point for a beta subunit off domain 1, and this is a covalent attachment, and you can either attach on beta 2 or beta 4, or of course you can attach on nothing. Okay, there is then also the option of attaching a beta subunit onto domain 4, which is a non-covalent interaction, and you have the option of attaching either beta 1 or beta 3 or nothing. Okay, right, so now we've discussed that, we're going to abandon the beta subunits now. We're just going to go back to focusing on the alpha subunit, which is the pore-forming subunit. This forms a voltage-gated sodium channel whether you put beta subunits onto it or not. These really just moderate where it goes in the cell. Okay, so let's now discuss the opening and the inactivation of voltage-gated sodium channels then. So I'm going to go back to this picture that I promised to go back to earlier. Okay, so we're looking now at the alpha subunit from above. Here is the pore down the middle, and here are the 4 separate domains. Okay, and I told you that at the center, each domain put in its P loop basically, which can also be called its pore loop. Okay, so we've colored these in red previously. Okay, there we go. And then around the outside of that, the other important structure that we have is this S4 membrane-spanning alpha helix, which you have in all 4 domains, 1 in each of the 4 domains. Okay, and these have got positively charged amino acid residues within them, and you have a positively charged amino acid residue every 3 amino acids, basically. So these are incredibly positively charged. Now, what I want to do is imagine taking a cross-section down like so that we see these S4 membrane-spanning alpha helices as well. That's the only reason that I've done it at that odd angle, so that we'll go through one of these S4 membrane-spanning alpha helices. Okay, so what you'll get is a picture that looks something like this. So here is the cell membrane, and then sitting in the cell membrane we have our voltage-gated sodium channel. Okay, and it will have a shape that looks something like this. Okay, like so. Then let's have the other one here. Let's put that in like so. So initially, the voltage-gated sodium channel that I'm drawing at the moment is closed. So we're assuming that the electrical potential difference across the cell membrane is at the moment resting membrane potential. So it's currently at negative 65 millivolts. Okay, and therefore our voltage-gated sodium channel is currently in the closed state. So this is a closed channel. Okay, right. So these little overhangs that you've got here at the top, these are the P loops. Okay, so this is this red portion on this picture. So before we were looking from the top, so we were looking from up here before. Okay, so there are the P loops. Then we've got our S4 membrane-spanning alpha helices sitting here, and we picked a clever cross-section so that we go through 2, even though I managed to miss it when I was drawing that cross-section there. Okay, so we've got these 2 S4 membrane-spanning alpha helices there. Okay, right, so these are our voltage sensors, and remember these are positively charged. Okay, right, so now let's discuss this P loop in a little bit more detail. So basically, when the channel opens, these portions here which are currently occluding the channel, they will move aside, and then things will be able to move through. And once those bits have moved aside, basically, this will be the narrowest portion of the pore. The pore will move back, basically. You can imagine it moving back to here. Once we're open, these will move out of the way and it will go back to there. And then this will be the narrowest portion of the pore. And this narrowest portion of the pore, which is lined by these P-loops, is known as the selectivity filter. And this is the portion that is responsible for making sure that the ions which actually move through this ion channel are sodium ions rather than just any old ion. So it is this which ensures that 10 times more sodium ions will move through this channel than potassium ions. Okay, so the channel is 10 times more permeable to sodium ions than it is to potassium ions, and it's thanks to this selectivity filter which is lined by these P-loops. These are what help to make sure that the ions which actually move through here are sodium ions rather than potassium ions. Okay, right. So, let's now discuss how the channel is going to activate, basically. So, initially, when the electrical potential difference across the cell membrane is at negative 65 millivolts, that means that the electrical potential intracellularly is 65 millivolts lower than the electrical potential extracellularly. Okay, these S4 membrane-spanning alpha helices, which we have 4 of in our alpha subunit, are all positively charged. So they're all going to feel a pull downwards towards the intracellular compartment, basically. So at the moment, when we're at resting membrane potential, all of these S4 membrane-spanning alpha helices are being pulled down towards the intracellular compartment. Now, if we depolarize the electrical potential difference across the cell membrane, let's draw this here. Okay, so let's say we take it up to threshold potential. So now the electrical potential difference across the cell membrane is up at negative 40 millivolts. Okay, right. What's going to happen? Well, now the electrical potential intracellularly is only 40 millivolts lower than the electrical potential extracellularly. So the pull that these S4 membrane-spanning alpha helices are going to feel is going to be reduced. So what will happen is they'll relax a little bit. They will move upwards, okay? So what's going to happen is they're all going to move upwards because the pull that they're feeling inwards has been reduced. They're still feeling a pull inwards because it's still, we've still got a lower electrical potential inwards than outwards, but the pull is no longer strong enough to keep them in this really pulled down position that they were in. So they're just going to spring back up, basically. And when they spring back up, they pull these portions that are occluding the pore down here. Okay, so these portions that I'll now highlight in orange, that I've drawn occluding the pore there, they will pull those out of the way. Okay, and I'll show this in brutishly blunt form by showing it like this. Okay, so what will happen is we'll now have something that looks like this, okay, which has completely opened the intracellular aspect of the pore. And now you can see that the P-loop lined portion of the pore, the selectivity filter up here, really is the narrowest portion of the pore. Okay, so this portion in red up here, this is the selectivity filter, and it should be pretty obvious now that that's the narrowest portion once the channel is actually open. Okay, and what's now happened is these S4 membrane-spanning alpha helices have moved up a little bit, and when they moved up, they've pulled these bits with them, and that's what triggers the conformational change, basically. Okay, now this then is the open state of the channel. This is the state which can conduct sodium ions, and this will allow sodium ions to come into the cell, and that produces the depolarizing upstroke of the action potential. Okay, so that's how we go from the closed channel to the open channel once we get to threshold potential. Okay, and that's how depolarization opens the channel. What we now want to discuss is the inactivation process, because remember, these voltage-gated sodium channels, they only remain open for around 1 millisecond, and then they close even though the electrical potential difference across the cell membrane is still hugely depolarized. Now that doesn't make sense from what I've told you so far, because if we're depolarizing the cell membrane, these S4 channels are going to remain pulled upwards basically, and that means that this bit should remain open basically. So why does it close? What causes it to close? Well, basically there is this concept of an inactivation gate. Okay, so basically there is this portion that dangles down, and I'll tell you exactly which portion this is. And I'm going to draw this in a way that people often do in cartoons, as a little ball and socket, well, ball and chain basically is what people call it. Okay, so this ball is attached to a chain basically, and it's not literally like this. I'll show you the actual structure in a moment. Okay, but this is how we can show it in cartoon form. Basically, dangling down from the alpha subunit, you have this structure which can basically lodge itself into the pore here. So at the moment it's not lodged in, and the channel is originally open. And of course we need to draw it on here as well. So here is this ball and chain, and of course when you were sitting in the closed form, the ball could not go into the pore because the pore was closed from the inside, so the ball couldn't get in then. But now we've opened it, and now this ball can go in. So basically this 1 millisecond is the 1 millisecond where you're open and the ball hasn't quite got in yet. And then what's going to happen very quickly after 1 millisecond is the ball is going to go back in there basically. Well, it's going to go in there for the first time. Okay, so here is our channel now, and this special ball-like domain has now stuck itself into this portion here and has blocked the channel. Okay, so this process of it going in like that, that's called inactivation. Okay, so you've now got a channel that is sitting in the inactivated form. So notice there is a difference between the inactivated channel and the closed channel, a very big difference. In the closed channel, the reason no sodium is coming through is that this portion of the channel here is occluding it. Okay, so the channel is closed from the intracellular aspect. Now that portion is no longer blocking the channel. Instead, this domain here, this globular domain, that's what the fancy name for this is called. It's called a globular domain, or even better, an inactivation gate, and I'll show you that in a moment. So this is a globular domain. Oh, damn. There's a globular domain. This is now sitting in the pore and blocking it. Okay, so I'll draw this globular domain in turquoise here. Okay, and this globular domain will go into the pore in about 1 millisecond after opening the pore, basically. Okay, so the fancy name for this globular domain is called the inactivation gate. Okay, and it's what closes the channel off after 1 millisecond. It takes the channel from the open state into the inactivated state. Okay, now the channel will stay like this until you repolarize the cell membrane. When you repolarize the cell membrane, when you take the electrical potential difference back down to, let's say, negative 65, what will happen then is under that heightened electrical potential difference across the cell membrane. So once the electrical potential difference across the cell membrane becomes nicely negative again, okay, so if you take the electrical potential difference back down to negative 65, the inactivation gate will come back out. So this will come out, and then of course you'll go back instantaneously into the open state for a moment, and then because you're back at negative 65, it will return back to the closed channel. So you can't go directly from the inactivated state to the closed channel. What you have to do is you firstly have to take the inactivation gate out, and then you'll be in this open state for a tiny transient period, but of course the S4 membrane spanning alpha helices will have moved back down because of the electrical potential difference being returned down to negative 65, and they will then cause the conformation to return back so that it's closed off down here once again. Okay, so this process of going out of the inactivated state is called de-inactivation. Okay, so activation is going from the open state into the inactivated state. Going from the inactivated state back into the open state transiently and then moving back into the closed state is then called de-inactivation. Well, this step here is called de-inactivation. Okay, so that's the basis of the inactivation of the voltage-gated sodium channels. They remain open for 1 millisecond, then the inactivation gate closes, blocks them completely, and the inactivation gate will only come out once you've taken the electrical potential difference across the cell membrane back down to negative 65 millivolts. And whilst the inactivation gate is there, you can't actually go back to the closed state. So until you've taken the inactivation gate off, you can't go back to the closed state. So firstly, you have to take the inactivation gate out. That will return you transiently, and I mean, instantaneously back to the open state, and then it will move back from the open state into the closed state. So basically, these domains here can't come and occlude the channel whilst the inactivation gate is in the way, even if you've gone back down below negative 40 millivolts. So you need the inactivation gate to come out before you can then close the channel. Okay? Right, so now let me tell you the real correlate of this inactivation gate. Okay, so let me get you some more paper and I'll show you the real correlate of it. So we need to look at the structure of our alpha subunit again so that we can see this inactivation gate. So let's draw this picture out because it doesn't hurt to revise. Okay, so here is our membrane. And remember, the alpha subunit of our voltage-gated sodium channel has these 4 domains. So here's the amino terminus, and then let's begin domain 1. So we have our 5 membrane-spanning alpha helices, then the p-loop, then the 6th membrane-spanning alpha helix, S6. Then we start domain 2. We have S1, S2, S3, S4, the p-loop, S6. Okay, then we've finished at domain 2, we're on to domain 3. We then have the first membrane spanning alpha helix, the second, the third, S4, S5, the p-loop, and then S6. And then we finally have domain 4 over here, S1, S2, S3, S4, S5, p-loop, S6, and then the carboxylic acid terminus of our alpha subunit of our voltage-gated sodium channel. Basically, the real correlate of the inactivation gate is this loop between the 3rd domain and the 4th domain. Okay, so this dangles down into the cytoplasm, and it is this loop that comes into the pore of the voltage-gated sodium channel alpha subunit from the intracellular aspect, binds, and then blocks the pore. Okay, so this is the real correlate of the inactivation gate. Okay, right, and there is an extremely important combination of 4 amino acids within this loop between domain 3 and domain 4, which actually is responsible for binding within the pore and blocking the pore so that sodium ions cannot bind. So basically what I mean is that this portion in turquoise is this ball, and I want to now show you the 4 amino acids which are extremely important for it being able to bind within the pore and thereby blocking the pore. Okay, so basically the sequence of 4 amino acids that this loop between domain 3 and domain 4 has, which is so important for this, is a sequence that in single letter amino acid code reads IFMT. Okay, so to be able to read that, you need to be able to obviously read single letter amino acid code. So let me help you with this. So basically that first amino acid is an isoleucine. Okay, so that's what I stands for. So that the first amino acid that points towards the amino terminus, that's an isoleucine. So let me show you the structure of an isoleucine amino acid, because I think it would be quite nice to actually draw out this sequence of 4 amino acids. So the first amino acid then, here is its amino terminus. Okay, then we've got its alpha carbon with a hydrogen coming off it. Then we've got the carboxylic acid domain. Okay, so that's the core amino acid structure. Then the R group of an isoleucine amino acid basically consists of 2 carbons, like so, then with 1 methyl group coming off this first carbon here, and then a second methyl group coming off this, a first methyl group coming off this first carbon here, and then a second methyl group coming off this second carbon here. Okay, and then we'll put hydrogens coming off everywhere else. Okay, like so. So that's the R group of isoleucine. Okay, the next amino acid along is F. Now, what does F stand for? So I stood for isoleucine. Next, let's have F. F basically stands for phenylalanine. And you might wonder, well, why didn't phenylalanine get called P? Well, P was for proline, so it couldn't have that. So they thought to themselves, well, what's the best letter to give phenylalanine? Well, it sounds as though it begins with an F, so let's give it F. So let's draw out the phenylalanine again. So here's the amino group of the phenylalanine bound to the carboxylic acid group of the isoleucine before it. Then we have the alpha carbon with a hydrogen coming off it, then the carboxylic acid group, and then the R group of phenylalanine consists of a methylene group, like so, then with a phenyl group coming off the side of it. And that's the basis for its name, because alanine is the amino acid when you just have a methyl group coming off. And because we've got a phenyl group then coming off this methyl group, it is therefore phenylalanine. Okay? So phenyl is just a benzene ring, basically. When you have a benzene ring attached to something as a group, that's called a phenyl group. Okay, so that's what F stands for. Next, we've got M. M stands for methionine. Okay, so where should I write this? I'll put it... no, I'll continue on up here. So M is for methionine. So the next amino acid along is going to be methionine. So here's the amino group bound to the carboxylic acid group of our phenylalanine. Here's the alpha carbon with a hydrogen coming off it. Here's our carboxylic acid group. And then methionine basically consists of 2 methylene groups. So here's one, then another, okay? And the fancy name for 2 methylene groups joined together is an ethylene group. Then you have a sulfur atom, and then off the sulfur atom you then have a methyl group, like so. Okay, so that's a methionine amino acid there. And then the final amino acid along is T, and T is for threonine. Okay, so this stands for threonine. And basically, threonine is very similar to serine. It has a methylene group with then an alcohol group on, except that you've added an extra methyl group in. So I'll show you this. And so here's the amino group. Here's the alpha carbon with a hydrogen coming off it. And of course I should just put the carboxylic acid group to complete the core structure, which will then be bound to the amino acid after it, somewhere down here. And then the R group of threonine consists of a carbon with an alcohol group coming off it. It also has a hydrogen coming off it, and then a methyl group as well, like so. So that's threonine. So this is this sequence of 4 amino acids that you have present in this loop between the 3rd domain and the 4th domain of our alpha subunit of our voltage-gated sodium channel, which is responsible for binding within the pore of the voltage-gated sodium channel alpha subunit, and thereby blocking the pore from the intracellular aspect. Okay, right, and that's the real correlate of that ball that I showed you earlier on. Right, okay, so that now that now finishes our discussion of the opening and the inactivation of the voltage-gated sodium channels. What I now want to discuss to finish with is certain toxins which work by blocking voltage-gated sodium channels. Okay, so there are two I would like to discuss. The first is tetrodotoxin. Okay, so this is quite a famous neurotoxin. This is present within pufferfish. Okay, so this is why you have to be very, very highly trained in order to cook pufferfish in a restaurant, because if you don't cook it properly, if you don't prepare it properly, you can actually end up with tetrodotoxin poisoning, and it can be fatal because it can cause respiratory paralysis. It can stop the diaphragm contracting. Okay, so a very, very dangerous toxin, and it's going to work by blocking voltage-gated sodium channels, thereby blocking action potentials from firing down the phrenic nerve, also blocking action potentials within the diaphragm itself, and therefore blocking contraction of the diaphragm, and you won't be able to breathe, basically. Okay, so tetrodotoxin is a very dangerous neurotoxin. Another example is saxitoxin, and before we just move on to saxitoxin, let me also add that tetrodotoxin is often abbreviated to TTX, T for tetrodo, TX for toxin. Okay, and the reason I'm not discussing the function yet of tetrodotoxin is that the function of saxitoxin is exactly the same, so we might as well discuss them together. Okay, so I'll just introduce saxitoxin as well, do the sort of general knowledge portion. Okay, so saxitoxin is often abbreviated to STX, okay, and this one is produced by dinoflagellates, which are single-celled protists that live within the sea, basically. And shellfish, such as clams and mussels, can consume these protists, and when they do, the toxin, saxitoxin, builds up within them. So certain organisms within the class of dinoflagellates can produce this saxitoxin. Now, dinoflagellates, as I say, are within the family of protists, or rather the kingdom of life protists. So remember, there are 5 kingdoms of life. There is Kingdom Animalia, which is animals, Kingdom Plantae, which is plants, Kingdom Prokaryote, which is bacteria, Kingdom Fungi, which is fungi, and then there is also the kingdom that no one can ever remember, which is Kingdom Protoctista, which is the protists. Okay, so there are a huge number of different strange things within this kingdom, and some of them are dinoflagellates. Okay, and these are basically single-celled organisms, okay, which live within the sea, generally. I think some of them can also live within fresh water, but most of them live within the sea. And basically certain shellfish, such as clams and mussels, and there are other shellfish as well, okay, can consume these dinoflagellates. And these dinoflagellates, some of them produce saxitoxin. So what happens is the saxitoxin builds up in the clams and the mussels, and the humans then eat the clams and the mussels, and they get saxitoxin poisoning. Now, you're particularly at risk of this if you eat clams at a time of year when the dinoflagellates are doing very well. Okay, so sometimes what can happen is you get a bloom of these things, and often that causes the colour of the water to change, basically, and often it causes it to go red, so it's called a red tide. Okay, so when these things do too well, they actually get so densely populated, they change the colour of the water to red, and I highly advise that you, if you haven't seen pictures of this before, that you Google it. It's quite dramatic. It looks as though the water's turned to blood. Okay, but basically, if you then eat shellfish which have been exposed to this massive population of dinoflagellates, you're in for trouble, basically, because the shellfish have been feasting on this huge population of dinoflagellates, and some of them might be producing this saxitoxin, so that shellfish have far too high concentration of this. You're then ingesting that, and it then causes neurotoxicity. So how does it cause neurotoxicity? Well, it's the same as tetrodotoxin. It causes paralysis of the diaphragm and also the phrenic nerve firing to the diaphragm, which then stops you from breathing. So that's how it kills you. But what's it actually going to do? Well, basically, both of these toxins bind on the extracellular face of voltage-gated sodium channels alpha subunits. So if I draw an alpha subunit here, okay, like so, basically what happens is both of these toxins can bind to the alpha subunit of voltage-gated sodium channels from the extracellular aspect, but they generally prefer the open state. Okay, so they are what are known as use-dependent blockers. Okay. And what this means is that they only generally bind to the voltage-gated sodium channels when the voltage-gated sodium channels are open. So, you will only find them bound generally when the voltage-gated sodium channel is open. Their affinity is much higher for the open state than for the closed state. Now, when the voltage-gated sodium channel opens, what happens is these drugs bind to the pore from the extracellular aspect. Okay, so here in pink, this is the drug binding to the pore from the extracellular aspect, and hopefully you can appreciate that what it will do is it will block that pore. Now, sodium ions are not going to be able to move through that channel. Of course, when the channel then inactivates, and then when the electrical potential difference goes back to repolarized level, and you undergo de-inactivation and you go back into the closed state, the drug will then fall off, basically, because it's use dependent. It only binds with a good affinity to the open state of the channel, but basically, by binding to the open state of the channel, it's going to block the movement of ions through that. And that means that you're not going to get action potentials being formed, because the instant the voltage-gated sodium channels open, these toxins are going to bind to the pore from the outer aspect, and they're going to block the movement of sodium ions through. So you're not going to be able to fire action potentials within your phrenic nerve. You're not going to be able to fire action potentials along the skeletal muscle of the diaphragm, so you're not going to breathe, and that's what kills you. Okay, moreover, these two toxins, tetrodotoxin and saxitoxin, they will bind to the extracellular pore region of all of the alpha subunits, pretty much. They have differing affinities for each of them, but they have a good affinity for all of them, basically, so they can block all voltage-gated sodium channel alpha subunits in this use-dependent manner. Okay, so that now concludes our discussion of voltage-gated sodium channels.